So first of all, thanks very much for having me here. It's great to be part of this and part of your opening inaugural. So I'll remember that as you become bigger and bigger, John. So that's fantastic. So I changed the title of this talk. So building better transcriptomic maps to build better therapies. And as John said, I think I got the tough call here about how you apply all of this. So I thought I'd start with the question of why we bother with maps at all. And partly this was an excuse to be able to put an Indiana Jones and the Raiders of the Lost Ark map up. But it does actually genuinely have some meaning beyond that. So I think in terms of why we bother with maps, I think we've heard a lot about different kinds of mapping during the day. I think we map because we want to navigate to new places, because we want to grasp the relationship between different areas of the genome often. So things very much like where you've got contact points between enhancers and promoters. We also map to try and help plan, to make decisions. And most of all, we map to find treasure. So treasure in this case is treatments. So, but why do I need a good map? As John said, I'm actually involved in the UK Dementia Research Institute, and I'm a clinical geneticist. So why do I need a map in clinic? I need a map for the same reasons. I want to know, I want to navigate to new places. I want to find variants that have not been found before that are pathogenic for patients. So that is variants often in non-coding genomes that haven't been, or non-coding regions of the genome which haven't been considered in the same way. I might also want to grasp the relationship between features in a map. So I might want to know what other genes or what other variants might actually cause the same disease. So I need that map. I also want to map to help make decisions. And what decisions do I want to make? Well, I want to be able to plan and make decisions for my patient. What is going to happen to them? Are they going to have a particularly poor outcome from this genetic mutation or a better outcome? And all of that depends on the map that they hold and our ability to interpret it. And most of all, I want the map to find the treasure. I want to know how to treat or cure my patient if I can. So I need a good map. Do I have a good map? I don't know that I do at the moment. So I think, as I think someone put earlier today, that genomic medicine depends on maps of genes and maps of transcripts. The actual transcriptomic map or other types of functional genomic map are only second in importance to the sequence in itself. And actually maps really matter. If it's not on the map, you don't know it's there. So if we look at the map of the world from 500 BC, as some of you might notice, there's no Australia, there's no North America. There are large portions of the world that aren't there at all. It looks really pretty identical if you look at the Mediterranean, though. So you can have maps which are patchy in quality, and that's what I would argue we have at the moment in terms of our transcriptomic maps, and probably in terms of our epigenetic maps or our enhancer maps and so on. They're patchy. And what we want to move towards is something that's a bit more like a 2024 map of the world, a satellite map, where we can see the weather systems. Maybe that's the epigenetics, that's the methylation marks, whatever it is you want. But that map has so much more detail on it, and so is so much more useful. So, is the current transcriptomic map good enough, though? Well, it might be good enough in some places and not good enough in others. So this actually relates to analyses that were run back in 2020, but I think they still are quite instructive. So what we actually did was look at how much of the transcriptome, how much of the short read RNA sequencing data was used here from the GTEx project to just really try and assess whether there was read density in terms of RNA sequencing reads over regions of the genome which were not supposed to be exons, so either intronic or intergenic regions. And what we found was that actually a lot of reads were located at levels which were like an exon was there, but no exon annotated, particularly in human brain. And in some ways that's quite predictable. Human brain is hard to sample. It's hard to get enough detail. There are lots of different cell types in it. And so actually, in a way, if we'd even studied fetal brain, we'd have probably got an even more detailed understanding that we didn't really know that transcriptome map terribly well. But of course, I'm somebody who's focused on neurogenetic disorders, and so this particularly matters to me. We also, in that same piece of work, tried to analyze whether or not that novel transcription in the genome was actually targeted to particular cell types, whether some cell types had less quality annotation than others in terms of their transcriptomes. And actually, at the time, I would have guessed that it was excitatory neurons, but I should have been wiser about it. The reality is that what we found was that we were often finding novel exons that connected through exon-exon junction reads to genes which are highly oligodendrocyte specific. And I think that's really because actually oligodendrocytes are the cell type which wrap around and myelinate neurons. They're very hard to isolate on their own. It's only in the last year or two that there are protocols to generate iPSC-derived oligodendrocytes. In fact, there are lots of different types of oligodendrocytes, probably multiple classes that go into continuum. And they're very, very difficult, as I said, to isolate on their own and to get good quality RNA out of them because of the high myelin content. So it's perhaps not so surprising that we don't actually know their transcriptome terribly well, and we're hoping to change that. It's also other areas of the genome, you might argue, or the transcriptome, I should say, are poorly understood. And that's just, again, because of the structure of genome itself. We've known for a very long time that if you have a gene and you have a pseudogene and you want to use short-read DNA sequencing to identify mutations in the main gene, so PKD1, for example, you have a problem, which is that lots of your short reads multi-map to the pseudogene, and you can't tell whether your variant is coming from the pseudogene or coming from the gene itself. And there is no reason why that does not occur in the same way for genes and pseudogenes and their transcripts. And that is what we've also looked into with some key examples. So how can we solve this? Well, we think that long-read RNA sequencing, both targeted and untargeted, is a way to navigate to new places in the brain transcriptome. And we focus so far on targeted, but we have been using untargeted as well. And the reason why it's so powerful is for the first time, you can go from one end of the transcript to the other. You don't have to guess what transcripts are being made by trying to piece together the expression of individual exons. And as somebody who actually used Affy exon arrays, I feel like that's a great thing. So I date all the way back to the exon array era. But you know, I think it's remarkable to be able to get that complete information and to have it actually with that much clarity. And there are two platforms that are allowing us to do this now, Pacific Biosciences and Oxford Nanopore. And we looked at a protocol that was actually adapted from something that was generated, first of all, by PacBio to generate targeted long-read RNA sequencing data off of targets of interest without necessarily having to know what the actual transcript structure was. So what we did was generate probes which were targeted to both exons and introns into the flanking regions of our genes of interest, or transcripts of interest, I should say, in order to capture transcripts without biasing it towards what we already know. So we've done that kind of work, and I'm just going to give you a couple of examples of where we've been able to navigate to new places within old genes. And so one example is here in GBA1, and it's pseudogene GBA pseudogene 1. So a bit like, I guess, SMN1 and SMN2, where SMN2 was actually for a long time considered a pseudogene of SMN1. And that is now, SMN2 is now a target for maybe the most successful antisense oligonucleotide currently on the market, nusinersen, for the treatment of spinal muscular atrophy. We actually wondered whether GBA1 and its pseudogene, GBA pseudogene 1, was GBA pseudogene 1 even a proper pseudogene? Could it make protein, for example? What do we know or what do we not know about GBA1 transcripts? And so we applied targeted long-read RNA sequencing to both these genes. And we identified novel transcripts which would be predicted to encode novel proteins. And actually, in the case of GBA pseudogene 1, because those novel proteins actually had novel peptide sequences that can be identified in mass spec data, we were able to demonstrate, using just publicly available mass spectrometry data from human brain, that in actual fact, the pseudogene can make protein, that there are transcripts which are translated and which make stable protein in the human brain, and which may give a substrate to new approaches to thinking about conditions which relate to GBA1 and GBA pseudogene 1. So that would be Gaucher's disease, which is caused by biallelic mutations in GBA1, and that would also be Parkinson's disease, where GBA1 in mutations in a single allele actually give a very much increased risk of Parkinson's disease, particularly with dementia. So here, what I'm showing you is actually the fact that we've got these novel protein structures of GBA1, which are essentially fragments of the full-length GBA1, and these are predicted with AlphaFold, and also the same sort of approach applied to GBA pseudogene 1. We've also taken targeted long-read RNA sequencing to look at another old gene, as it were, APP. So For those of us in the neurodegeneration field, APP is incredibly important. It encodes for amyloid precursor protein. When this gene is mutated, it gives rise to an early onset familial form of Alzheimer's disease. And in actual fact, mutations of this gene were the very first mutations to be identified and capable of causing early onset Alzheimer's disease. In a sense, they really changed the field and started a race for more genes. At present, the way we understand how this gene generates Alzheimer's disease is the idea that it encodes this amyloid precursor protein, which is cleaved at two positions, with a beta secretase and gamma secretase to form A beta fragments, which are then very pro-aggregation and which actually aggregate and form the amyloid plaques that are classical for Alzheimer's disease. What we identified when we did long-read RNA sequencing of APP was in actual fact there are transcripts being made from APP which are not full length and which in actual fact are very, very closely aligned to the beta secretase cleavage site. And so in actual fact, they mean that you do not need to have beta secretase activity in order to generate fragments of A beta, which can actually form amyloid. And so you have an independent pathway to amyloidogenesis, which does not depend on beta secretase activity. And in actual fact, in human brain, we found that these novel transcripts, which encode for a C100 amino acid fragment of APP, are actually present in percentages in human brain of around 4%. So quite enough to actually be sufficient over a lifetime to be important, given that actually only a small quantity of APP protein is actually required to form A beta to, in fact, give rise to familial Alzheimer's disease. So you only have to wait by those kinds of percentages to actually get an early onset AD. This work is on bioRxiv. It's just been posted in the last month. What I was going to focus on, though, in this was actually to look at how you target these things for therapies. And this is one place where we've started that in the realm of Parkinson's disease and focusing on another old gene, synuclein. So synuclein has a long history in Parkinson's disease. It was identified really to be the major constituents of Lewy bodies, which are pathognomonic for Alzheimer's disease and are the key pathology. And in actual fact, it was found in 1997 that, as I said, that SNCA encodes for the major constituents of Lewy bodies, and also that single-point mutations in the gene give rise to early onset PD. We also then found that multiplications of SNCA, so copy number variants, duplications, triplications of this gene, also give rise to familial Parkinson's disease with a high psychiatric component attached to it. We then found in 2012 that common genetic variation around this gene also gave an increased risk of Parkinson's disease. And as you can see on that classical plot of frequency of alleles in populations versus risk of PD, and you can do this kind of plot for lots and lots of diseases. Actually, the interesting thing here is that synuclein is important for very rare familial forms of Parkinson's disease, but it's also very important for as being an important risk locus for common variant risk. So this is a particularly important gene because actually it not only contributes to the rare forms, but to the complex forms. And we know that both synuclein dose and type impact on alpha-synuclein aggregation and impact on disease risk. So what did we do? Well, we looked at SNCA transcripts in dopaminergic neurons. So for those of you who aren't versed in Parkinson's disease, the first symptoms of this disorder, and the ones that prompt diagnosis, are the motor symptoms. And these motor symptoms are classically bradykinesia, tremor, and rigidity. And they arise from the death of dopaminergic neurons, which actually have to drop by 80% in number to actually cause the symptomology. So these particular type of neurons are very, very important. So we actually used iPSC-derived midbrain dopaminergic neurons and looked at these neurons in control, SNCA triplication, and A53T cell lines. We then used this targeted long-read approach where we actually designed lockdown probes across the introns and exons of this gene to actually try and pull down all the transcripts from SNCA. So what did we find? Well, we actually found a lot of transcripts that we were not expecting to find. So this is, of course, a very well-studied gene. We shouldn't really be finding anything, is the reality. It's already being targeted for therapeutics in trials and even ASO trials at the moment. So we should find zero, in fact. But what we actually found was novel transcripts, some of which would predict novel coding sequences, and many which would predict novel 3' or 5' UTRs. The other thing that we found, and this is on the second plot here, is that actually some of the transcripts that we were finding, these novel transcripts, are actually used remarkably commonly. So you might go, look, this is an 80% pure cell culture. This is all neurons, and most of them are dopaminergic neurons. So there should be one major transcript of this gene, and I shouldn't be finding much else. In actual fact, when we rank transcripts according to their usage, and that's what we're doing on the x-axis, and then the percentage usage of a transcript relative to all transcripts generated from that locus, we see that in actual fact the most commonly used transcript is only being used at around 25%. And we find that the fifth most common transcript being used is actually predicted to encode a novel open reading frame. And this is being done with very high confidence. We were actually very conservative. So we have approximately 100,000 full-length reads per sample. We needed a transcript to be detected in all samples to call it, with a minimum usage of 0.3%. So we actually have a huge amount of support for each of these transcript structures, equivalent to around 300 full-length reads on average per sample. And there were over 40 samples in this data set. So what are the implications of finding novel transcripts of SNCA? Well, one implication might be around SNCA aggregation properties. So I've told you that alpha-synuclein is particularly important because when it aggregates, it forms these Lewy bodies, which are actually the key feature of Parkinson's disease and which are thought to be critical for the pathogenesis. We also know that we might be interested in synuclein because synuclein is actually being currently focused on as a target in terms of lowering its levels, lowering its mRNA levels, using antisense oligonucleotides and other RNA targeting approaches. So most commonly around thinking about using ASOs to actually have RNA's H-dependent degradation of synuclein RNA. So these novel transcripts, we focus on the novel proteins first. So actually what we found was there were changes primarily at the 5' end of the gene, which resulted in novel amino acid sequences. So that is novel in terms of the length of the sequence. So classically, synuclein would have 140 amino acids in it. That would be what most people expect from most of the transcript structures. But you do actually have these predictions of shorter isoforms of synuclein, that might be 113, 118 amino acid sequences, amino acids long, and that actually they have unique peptide sequences within them. So they're not just truncated forms, but they actually have a unique runs of amino acids. So that allows us to do something, which is to, again, hunt through the public mass spec data and ask ourselves whether actually these are translated. We could also do a targeted approach where we actually took human brain samples from our own collection. We looked at substantia nigra and we looked at anterior cingulate cortex. We actually then selected for synuclein protein using an antibody approach and then ran a kind of targeted mass spec in effect. And we were able to demonstrate for ourselves that indeed we could detect synuclein isoforms that would be predicted to be 113 amino acids long or 118 amino acids long. And that these forms of synuclein are actually predicted to have lower solubility and to be more aggregation prone. And you might say, well, okay, does that really matter? Because they actually constitute small percentages of the total amount of transcript being made. Well, they might matter because this is actually a kind of seeded process. The belief is that small quantities of abnormal protein would be sufficient to actually cause a seeded event, which actually where more synuclein of a normal type might aggregate around it. And there's actually a theory of a kind of prion-like approach to Lewy body pathology. So this actually could be much more important than it looks just by the numbers alone. So then let's think about whether it matters from the point of view, these novel transcript structures, from the point of view of ASO targeting. And here, what we might think about is UTR variability. Because if you're going to actually generate antisense oligonucleotides, a lot of people are focused either on generating them against the 5' UTR sequence or the 3' UTR sequence, where there's a lot of variability which might be specific to that gene. So, for example, synuclein has actually, there's SNCA, but there's also SNCB. So you might want to think about targeting in places which are most unique. And the UTRs are often those locations. Now, what we found was actually that there is a high level of variability in 3' UTR sequences being used in the dopaminergic neurons. And in actual fact, if we look at the different sizes of those UTRs, you can see them classified here. This was work, very nice work done by Emil Gustafsson, who's also here. and also look at the relative usage of those 3' UTRs. And you can see that they vary in terms of how much that particular UTR is being used, and therefore what would be the value of targeting that UTR versus another. And so if we actually designed antisense oligonucleotides going across that UTR space and used them in our cell model systems. And what did we find? Well, we found what we predicted, actually, from the sequencing data, which is an antisense oligonucleotide that targets the vast majority of those UTRs will be the most effective at lowering the synuclein levels, both at the mRNA levels and at the protein levels. And what's the outcome of the usage of that? Well, if we look at the SNCA triplication models, where you have excess synuclein protein, when you use the right ASO, you can drop the protein levels, you can drop the levels of aggregation of alpha-syn, and you can drop the levels of cell death. So here what we have is an approach which we think, actually, if you expanded this, you could create a much more efficient approach to ASO design by understanding the transcript structures that you're trying to design against better. So it's a little bit like saying, if I was a small molecule person and I was doing small molecule drug therapeutics, would I really try and make a small molecule against a protein which I hadn't got a cryo-EM structure for example? Wouldn't I want to know the exact structure of that protein? So why wouldn't I, in an RNA targeting approach, want to do the same? So again, you might think of using that also for the purposes of making it cell type specific. So here, what we're showing here is in actual fact, the UTR usage varies between the different cell types. So looking at astrocytes, either treated or untreated with activators, microglia, either treated or untreated, and we could then, in effect, make an ASO that would be targeting both cell state and cell type. And that's something that we're interested in doing. So just to end on the point that, again, I think genomics has a massive capacity to transform the treatment of many diseases, including neurological diseases, but we actually need to have accurate maps to be able to do that, and that's all types of functional genomic mapping. So finally, just to finish on acknowledgements here and to thank people. So the biggest thing for my lab is thanking the people who donate samples and brain tissue for research. That's massively important to us. I also want to just highlight people from my own lab, in particular Emil Gustafsson, who's here, who's really driven a lot of the long-read sequencing work, but also James Evans and Charlie Arbour, who've been really instrumental on the iPSC-derived work for APP and for SNCA, and many others across. Thanks very much for listening.